In this video, we are going to look at how gene expression determines cell type. So as a review, remember, DNA codes for RNA, RNA codes for proteins, proteins make your traits. Now, how a gene becomes a trait is called gene expression. So a gene is expressed when it becomes a trait. And this process is going to be regulated by proteins, because again, proteins do pretty much everything in the body. Now, remember, in a multicellular organism, we have lots of different cells. However, all of our cells have the same genome, meaning that they have the same set of chromosomes and the same set of genes. Now, also in many multicellular organisms, we have what are called specialized cells. And these are cells that have specific structures so that they can accomplish specific functions. For example, nerve cells help transmit information. Muscle cells contract to move the body. But in all of these cases, these cells have the same DNA, and yet they have extremely different structures. So how do we get such different cells as nerve cells and muscle cells with the same genome? And the answer is that every cell doesn't use all of its DNA. A process called cell differentiation, meaning how cells become different, occurs where different cells are going to turn on and off the different genes that they need. So another way to say that is cells are going to express the genes that make the structures they need to accomplish their function. So if you think about it like a cookbook, DNA is a cookbook that has all the recipes, but it has all the recipes for all the cells. In other words, the DNA has the genes to create the structures, the traits, for all the different cell types. So each individual cell type is only going to use the parts that it needs to create the traits that it needs for its function. And how this works, again, is going to be completely regulated by lots and lots of different proteins. So for example, if we look at a red blood cell, a red blood cell does not need many of these genes. It has these genes in its genome, but it only needs to express of these hemoglobin, because hemoglobin is the only one that it needs to accomplish its function. And so it's going to not express or turn off the other genes. Stem cells are cells that haven't differentiated. In other words, they haven't specialized, so they have no specific structures, and you can think of them kind of like blank cells. They come as embryonic or adult stem cells. Now, embryonic are unique because they can become any type of cell. They have the ability to turn on and off any genes, and they also can build organs and things like that. So an embryonic stem cell could become any of these types of cells. And that's important because remember, at one point you were a single cell, a zygote, with one set of chromosomes, and from that, you had to differentiate and divide to create all the different specialized cells needed for a human being. So even though that one cell had just the one set of DNA, because it's an embryonic stem cell, it's able to turn into all the different types of cells that you need. Adult stem cells, which occur very, very early on in life, before you're even born, you stop having embryonic stem cells, they can only turn into one type. So you have some that make blood cells, some that make muscle cells, some that make nerve cells, but they can't make any of these. Now, embryonic stem cells can be really useful because since they can become anything and they have the code to build stuff, you could actually regrow entire organs for people. You could regrow a limb by using embryonic stem cells. The problem is embryonic stem cell use is fairly controversial because, again, they're embryonic stem cells, so they have to come from an embryo. However, we have come up with new ways that can potentially turn cells back into embryonic stem cells, and then we can use them to do all of those cool things that embryonic stem cells could do for us. Now, gene expression sometimes goes wrong. And because it's regulated by proteins, if you don't produce those proteins in the right way, it can impact how the genes are expressed. So too much, too little, or the wrong time of producing these proteins can also cause the gene to be expressed too much, too little, or at the wrong time, and cause a lot of issues. One of those issues is cancer. So cancer happens when certain genes and cell division are incorrectly regulated, and so they get out of control. So in cancer, the wrong genes are expressed, or they're expressed too much, and it causes uncontrolled division of those cells. And we call those tumors. And tumors can come into two types. Benign tumors are ones that don't spread. They might get bigger, but they don't spread to other parts of the body. Malignant tumors, these are the ones that are cancerous. And these are ones that can spread to other parts of the body, which is why you could start with this one kind of cancer and end up with a bunch of different types of cancer.